Now we're going to move to the membrane-bound organelles. Membrane-bound. So of these membrane-bound organelles, we have the endoplasmic reticulum, the Golgi apparatus, sometimes it's called the Golgi complex. Okay. We have these things called lysosomes. Lysosomes themselves. And we have the peroxisomes. And then we have the mitochondria. Okay. So we're going to start with the endoplasmic reticulum and the Golgi because they are very closely related. All right, so the ER, the endoplasmic reticulum. I'm just going to call it ER, the endoplasmic reticulum. Okay, so it's nothing more than a series of chambers and what it does, it is for storage. Okay, it is for the synthesis or detox. No, let's just erase that and start over. Synthesis or detox of substances. Okay. Now the individual chambers, these are called cisternae. Let me make sure I spelled that right. C-I-S-T-E-R-N-A-E, cisternae. Okay. So if we take a look at it, so here's our cell. So we're going to draw a very big cell right here. So here's our cell, and we're going to draw our nucleus a little bit smaller itself. Now attached to the nucleus, we've already said earlier, actually I want to do it like this. So the nucleus you're going to see is a double membrane. It's got two membranes on itself. And attached to the outer membrane, you have this kind of structure. Now this is just a poor artist rendition, and it's not like this at all. I mean, you'll have some chambers over here, some coming over here like this. So it's not all exact and beautiful, but that's what the endoplasmic reticulum is. It attaches directly to the nucleus, to the nucleus itself. Okay, so there are two basic types of endoplasmic reticulum, and let me label this. Here's our nucleus. So that's our nucleus, and this is our endoplasmic reticulum. Okay, the red guy, or the red thing. That's better. Okay. So there are two main types. We have what's called the smooth endoplasmic reticulum. Okay, and I said earlier when we were talking about ribosomes, when ribosomes don't attach to the ER, it's smooth ER. And this is for the synthesis of lipids and carbohydrates. Synthesis of lipids and carbohydrates. It is for the synthesis of cholesterol and phospholipids. Okay, I think that's just redundant. I'm sorry I did that, but it doesn't matter. Okay. So synthesis is real. It's also for the storage of cholesterol and phospholipids it is for the storage of steroid hormones okay, our steroid hormones are stored there, our glycerides are stored there, and glycogen will be stored in there as well. Okay. Another thing that's smooth endoplasmic reticulum for is it will be for detox itself. So like alcohol is detoxed in the liver, and the smooth endoplasmic reticulum of the liver is specifically designed to detox a lot of substances there. Okay, so that's the smooth endoplasmic reticulum. Okay, so then we're going to have the rough ER. Okay. So with the rough ER, we already know it's got a ribosome covered surface. Okay, ribosome covered. Okay, it is protein and glycoprotein synthesis. Okay. It's here that you get special folding of protein structure. Okay. And if a protein or something is made inside of the endoplasmic reticulum, okay, they're packages, products into vesicles. Okay, so let's take a look at our cell again. So if I just draw some dots on here like this, just random dots. Okay, so what we see here, where these dotted places are, that's our rough endoplasmic reticulum. Okay, so these areas right here, that's our rough ER. And then the non are where we don't have our ribosomes. Okay, so that's what the endoplasmic reticulum does. Okay, closely related to the function of the endoplasmic reticulum, we have what's called the Golgi. The Golgi, and we're going to put Golgi. The Golgi. Okay, it's the apparatus or complex, and basically what it does is it processes ER products. Okay. That's its job, just to process it. And so what those processings are is modifications to the products. So if you have a protein that needs to be changed a little bit, the Golgi will be doing that. Okay. And it's also for renewal of the plasma membrane. Okay. And it packages enzymes and proteins in vesicles for secretion. And secretion just means release from the cell. Okay. So if we go back to our cell here, So the Golgi, I'm going to draw it in this bluish color. The Golgi just looks like a bunch of flat pancakes. Okay, so if you look at the Golgi, looks like a bunch of flat pancakes, one right after another. Okay. So we'll be facing the other direction. Okay. So this is the Golgi. And so what happens? What happens is, let's say that the endoplasmic reticulum has finished creating something, and what it's going to do is going to release it in a vesicle. Now this vesicle is made of membrane. It's made of membrane. It's the exact same membrane that is on the cell, around the cell. It's plasma membrane. Okay, and what this membrane is going to do, okay, it's going to go and it's going to move, being carried by microtubules and microfilaments, intermediate filaments, and it's going to move and it's going to eventually fuse with the Golgi. And when it fuses with the Golgi, we're just going to say we're just going to draw some stuff in here. Let's just say some kind of protein or something. What that protein is going to do is it's going to move into the Golgi. And what the Golgi is going to do is it's going to process it. It's just going to do what the Golgi does to it. Okay. And then that's going to bud. Oops, wrong color. That's going to bud here. And the products are going to go into here. They're going to flow into here. And it's going to do some more modifications to it. And it's going to bud off. And it's going to go into the next one. And it's going to go into here. And then it will bud off. And then the products are going to go into here. And it's going to change it again. Eventually it's going to bud off. And then it's going to go into the next group. It's going to go into here. And then finally it'll be finished. And then what's going to happen is it's going to bud off again. And you're going to get new products. And they're going to move out this way. And eventually what's going to happen is they're going to bud to the cell membrane. And those products are going to be secreted. They're going to be secreted outwards. They're going to be let off and do what they need to do. So now when we say that it renews the cell membrane, this is actually the kind of the kick-ass part of it. Okay, so you remember we talked about this thing called endocytosis. Endocytosis. In endocytosis, what you do is you take part of the membrane here, and you actually lose part of the membrane when you make that vesicle with endocytosis. Now, if you didn't replace that, the cell membrane would actually get smaller, right? So what the Golgi does is when it packages these products out, this vesicle fuses with the membrane and it adds to the membrane size. So while endocytosis may pluck some of the membrane to make a vesicle, the Golgi is going to replenish that by taking a vesicle that it creates and is going to recover the loss. So that's where the actual replenishment of the cell membrane happens. Okay, so there are two sides to the Golgi. You have the cis and you have a trans face. So the cis face is on the same side, and then the trans is on the opposite side. So the cis face faces the nucleus, the trans face of the Golgi faces away from the nucleus. I don't think that's necessarily important that you remember that, but some people make it important to know that there are two sides to the Golgi itself. So endoplasmic reticulum, Golgi apparatus. Okay, basically they're there for making proteins, making proteins, lipids, detox, packaging things to be released from the cell. And the ER is also there for storage. So basically those are the roles of these two organelles. Okay? And I think instead of just talking to talk and make it longer, I think we'll just cut it off right there.